Bacteria, ideal organisms for investigating the molecules of life. This viscous material is made up of one of life's most famous molecules, DNA, the master molecule that provides the operating instructions for assembling a cell's proteins and other gene products. All life, from bacteria to mammals, develops and operates from a DNA blueprint. Macromolecules can be visualized through the techniques of gel electrophoresis, a process that separates them, revealing their overall structure and size. Colored dyes can be used to illustrate the principles of electrophoresis. These dyes are themselves macromolecules. To visualize just how big these macromolecules are, consider a molecule of water. An atom of oxygen bonded with two atoms of hydrogen. One hydrogen atom has a mass of 1 Dalton. The smallest macromolecule that can be imaged by gel electrophoresis has a mass of about 1,000 Daltons, or 1 kilodalton. The dye molecules will separate just like DNA and protein fragments. Each molecule has a mass of thousands of kilodaltons. The molecules carry surface charges, some positive, others negative. How these molecules separate on a gel depends on both properties, mass and charge. Positively charged macromolecules will be electrically driven towards the negative side, negative ones towards the positive side. The gel itself exerts a frictional resistance, acting as a molecular sieve to separate the moving macromolecules. Various dyes and a dye mixture show how this works. Energize, and the molecular race begins. The smaller molecules will migrate faster and so travel farther. Turning off the power will stop the molecular migration. The dyes have separated as predicted, with the smallest macromolecules racing ahead and the largest barely making it off the starting line. The same procedure can be used to analyze biological molecules such as DNA. These experiments begin by running fragments of DNA precisely cut by restriction enzymes. These fragments are of known length and are called marker DNA. A marker DNA allows you to plot a reference curve based on the known lengths of the marker fragments and their distances of migration during the run. Points for the curve are determined by the known base pair lengths of the marker fragments plotted against the distance run in millimeters. To find the length of an unknown DNA fragment, measure the distance run and read its length in base pairs off the reference curve. The type of macromolecule you wish to separate will determine which gel substrate to use for electrophoresis. The material used to separate macromolecules having a mass greater than 10 kilodaltons is agarose, made from seaweed. Agarose gel has a relatively homogeneous pore size, which makes it particularly useful for separating elongated macromolecules, such as nucleic acids. The appropriate gel to use for separating protein molecules is polyacrylamide, which is similar to the material used in soft contact lenses. Polyacrylamide gels are produced in precast cassettes. This precast gel is actually a gradient of macromolecular sieves that separate molecules in the 5 to 200 kilodalton range. Polyacrylamide Gel Electrophoresis, P.A.G.E, is the separation method used for polypeptides and proteins, including enzymes. The polyacrylamide used for gel electrophoresis forms a gradient of macromolecular sieves that separate molecules in the 5 to 2,000 kilodalton range, making it the gel to use for separating proteins, polypeptides, and enzymes. Ward's P.A.G.E tank is capable of running 1 or 2 8x10 gels simultaneously. The gel chamber has an inner and an outer compartment with gaskets which prevent the buffer from leaking into the outer compartment. There are 2 platinum electrodes, the cathode, located at the bottom of the inner compartment, and the anode in the bottom of the outer compartment. Prepare the concentrated running buffer solution. A working solution is then prepared by diluting 1 part concentrate to 3 parts distilled water. A glass cassette contains a precast gradient gel along with a 12-well comb. Choose the appropriate gel for the molecules you wish to separate. Remove the comb from the gel. Place the gel into the outer chamber, making sure the wells are facing in towards the inner compartment. Insert the wedges, making sure the gels are snug against the gasket to prevent leaks into the outer chamber. Fill the inner compartment with working buffer solution until it covers the wells. Using a pipette, flush out any air bubbles still trapped in the wells. Slowly fill the outer compartment with working solution to approximately the same level as the inner compartment. Using a micropipette, load your sample into each well. Prepared protein samples contain bromophenol blue, a tracking dye, and sucrose, which helps keep samples in their wells. Make sure all connections are dry. Attach the lid to the chamber, making sure it is aligned properly. Plug the patch cords into the power supply. Set the voltage to 125 volts and turn on the unit. When the power is turned on, gas bubbles will begin forming along the platinum electrodes. When the tracking die is about 2 millimeters from the end of the gel cassette, turn off the power. Remove the wedges then the gel cassettes. Carefully cut the taped edge of the cassette and open the cassette like a book. Carefully remove the gel from the glass cassette and place it into a staining tray. Add enough stain to cover the gels and allow to stain for approximately 45 minutes. Decant the stain and destain the gel until the background is clear. To enhance the banding, place the gel in white vinegar or 5% acetic acid. After the destaining, place the finished gel in a self-sealing bag for examination on the light table. The bagged gels can be stored refrigerated indefinitely.